Okay, first shot with DBS.
I'm going to practice serve.
Okay.
Because normally I can't really
Just
do an
about
overhead serve.
bionic serve coming my way.
Don't build it up. Right, here we go.
Parkies in a Pod is powered by Parkinson's UK, the charity that's here to support every Parkinson's journey every step of the way. Wherever you're from, whatever you need, visit parkinsons.org.uk for more information.
Hello, welcome to this stunning edition of Two Parkies in a Pod. We're centre court, Tooting Common at the moment.
Centre court, not Wimbledon.
No.
Tooting.
Tooting Common, yeah.
There's no audience or crowd, thankfully for us.
In the council-run courts.
In the council-run
But I have got a bit of Wimbledon here, actually, Kuhan, because this is your first game since DBS.
Are you marking your kit? Oh, Wimbledon balls.
Wimbledon balls I've got. So,
Official Wimbledon ball.
yeah, these were used at Wimbledon.
By?
I don't know.
Oh, is this the stuff that goes on sale afterwards? Or?
Yeah, we've got 3 of these.
Yeah.
And maybe they were used by, Radicano or something like that. Maybe. And we can dream, can't we?
Maybe they weren't used by anything.
A couple of ball boys used them as a knock-up, yeah.
Yeah. Oh, so it's not one of your Jurgen Klopp stories, is it?
No.
You said to me once, you've got a pair of Jurgen Klopp's football boots.
I really did, yeah.
Yeah, and I thought you actually meant like Adidas had did a partnership with him and you'd got one of his branded boots, but he donated his boots to you, right?
Yeah, my mate used to do his boots, so he nicked a pair for me.
Oh.
Keep it quiet, kid. Don't tell anyone.
Right.
Little plastic bag with Jurgen size 13 on it. Big feet, Jurgen had.
But more importantly, how are you feeling? Because this is our first game since the big D-Day for me at least. So
I'm feeling a bit intimidated by you, actually, because you're looking great, and I'm worried the Bionic Man's going to beat the...
Well, yeah.
You're the Steve Austin of tennis now.
Go on, explain Steve Austin. Explain
Steve
Bionic
Austin
Man.
was the $6 million man, and he was a test pilot. Lee Majors played the part.
Anybody younger than me, this won't make sense at all, actually.
They called him the $6 million man.
Yes.
And he crashed and they rebuilt him. $6 million seemed a lot of money in that time. My wife used to be a big Lee Majors fan, and she used to have a poster of him on the wall. And when he grew a moustache, she used to pencil in a moustache on him. And when he shaved off, she'd rub it out. So.
Really?
You're going to kill me for telling that story.
Excellent.
I'm a bit worried that you're going to be better than me now.
I'm not sure.
At tennis.
Dave, I'm not sure. Because I haven't moved. I'm the only person I know that spent 2 weeks in hospital and actually came out heavier than they went in.
That's because of all the ice cream you had.
Yeah.
920 ice cream doesn't help your figure, you know.
No, but needs must. Sorry, NHS, but the hospital food was terrible. So. Yeah, I ended up putting on a couple of kilos.
Well, I haven't played tennis for ages, so I had one game when you were off.
Yeah, who did you play with? Your sons?
I played with my old boss from Sky.
Ah, yeah. And did you win?
We won all.
Oh, right. All right.
I went off for the seconds, but it's. I'm thinking, hope it doesn't ruin it, everything, and you become really good. 140 mile per hour serve. I feel like it's a bit like me having a Dunlop Max Ply wooden racket, and you having a Rafa Nadal graphite shafted. Amazing.
Is it like who was that? 70s, the Swedish player.
Bjorn Borg.
Bjorn Borg versus
It was Donne.
Yeah, and the kind of wooden racket, and versus like a high-tech young, what's his name? The Spanish guy.
Rafa Nadal.
No, the new Spanish guy. No, the Italian guy. No, the Spanish guy.
Alvarez,
yeah. Are you drawing that sort of comparison?
I'm thinking we better get on there and have a knock around, see how we go.
Well, before we do, can we leave our listeners with the episode we did at the Brain Bank? Do you want to just let them know what we were up to?
Yeah, we had a great day, didn't we? We went to the Imperial College
yeah.
Department of Brain Sciences and the Parkies UK Brain Bank. It's quite an amazing place. You can donate your brain for research, and we went to see a brain being dissected. So here's what happened. Amazing day, and these are the guys we met.
So my name is Rory Turkman-Smith, and I'm the Tissue Bank Engagement Manager here, looking after both Parkinson's UK Brain Bank and the MS Society Tissue Bank.
Hi, I'm Steve Gentleman. I'm Professor of Neuropathology, and I'm the Scientific Director for the Parkinson's UK Brain Bank.
That's quite an impressive title.
Rory, just tell us about where we are and what happens here.
Of course. At the moment, we're on the 5th floor of Burlington Dames at the Imperial College Hammersmith Hospital campus, and that's where the brain bank is located. What we do is we enable people to donate tissues to research, and that's to support research into Parkinson's.
By donating tissue, you mean donating brains, right?
I do, but not exclusively. On the Parkinson's side, we're named the Parkinson's Brain Bank. On the MS side, we're the Tissue Bank. And that's to sort of show that we know both the donation of brain and spinal cord tissues.
How do you go about donating your brain here then?
Well, it's much like everything else in the health environment. There's forms involved. What I normally like to stress at this point is donation to research is different to what people might have heard about with NHS organ donation or, say, whole body organ donation. They're different schemes for different purposes. And so for us, it's a different set of forms which need to be completed, one, by the person who wants to donate, and secondly, by a next of kin. That's a dual role there because obviously when someone passes away, we'll need someone to communicate with us to inform us of that, and that will be their next of kin.
Just this morning, I was talking to my partner, Helen. I said, shall we sign up for this? And she's like, well, I don't need to because I've already got a donor card. And I had to point out it's a separate process, as you quite rightly described there. So just to be clear, the NHS donor card in the UK doesn't include brain donation.
That's correct. And thank you for pointing that out to your partner, doing the work for us. So in that sense, NHS organ donation is not exclusively, but is mostly focused on that live transplant aspect. You pass away, you may have a good kidney, someone else needs a kidney, they can make that work. There won't be so much clinical data release in there, which is very important for research.
Whereas there's no concept of a brain transplant, right? Not yet, right?
Not yet, no.
I've heard, Steve, that actually there was once a surgeon that transplanted a monkey brain. Have you heard of that case?
I have heard of the case. I don't know the details, and I'm not terribly sure of the outcome.
Ethically, probably not ideal, actually.
We're far from that, aren't we?
Well, we are. I mean, philosophically, if you think about a brain transplant, who survives? The body or the brain?
Yeah.
It's quite a philosophical nightmare.
Well, I had a philosophical debate with Helen this morning as well about consciousness and where it resides.
Quite the mornings you're having there, Kuhan.
I just said to have cornflakes or shreddies.
It's the kind of romantic conversation we have, But her point was consciousness was the whole thing. The mind, the body, and everything connected. But what do you guys think?
Steve, you first on me.
I knew it was coming to me. Consciousness is something very difficult to define. I spend my life looking at brains, which are physical objects, and I've been looking at those for 30 years trying to work out what's gone wrong. But consciousness is clearly not there. So where's it gone? I still really, after all this time, don't understand how this object is allowing you and me to talk to each other. So there's something else.
Something else, yeah.
Yeah, I mean, for me, on the sciencey side, I just always assume something in evolution has said it's a good idea to convince us that, we can interact with our surroundings and this is real, when a lot of it is just perception. It's input in, you're making it all up there.
Yeah.
I think it's probably separate to what is there.
Yeah. I interrupted you. You were giving your pitch about the brain bank. Do you want to continue? Because I just interrupted you.
No, of course. It was a nice segue. But yeah, and so I was sort of talking on those forms there, and that sort of third form to sign up is a little bit about someone's background health. And so that helps us inform basically things about maybe the first symptoms they had at diagnosis, if they have any other sort of clinical symptoms or sort of comorbidities and that sort of thing. And with that information, we're then able to basically inform the donation when that time comes. But similarly, we keep in contact with people about every 2 years with sort of follow-up questionnaires, see how their symptoms are going, see how their lifestyle habits are going, and that sort of thing. And that starts to build a very long, detailed picture about someone's life, which can then help us interpret what we find in the brain. Or not so much what I find in the brain, but what Steve finds in the brain.
What do you find in the brain, Steve?
All sorts of things. And I think it's important to realise that everybody's brain is different. Your brain is as different as you are in life. So every brain I look at is its own separate challenge to find out what went on. And what I'm really looking at is the culmination of all the changes that happened just during someone's life. So I can find all the pathologies, anything that's happened may be recorded or essentially in their brain. So that's what we do. We have a look down the microscope, what changes have occurred and what kind of symptoms might that have produced in life. What was the reason for that symptom?
Interesting. And just to the layperson, to the naked eye, does every brain look different as well?
I think most people have seen pictures of brains with all its folds on the outside. That's the cortex. That's what makes us human. And although the pattern's generally similar, it's about as individual as a thumbprint.
Mm. And you've had a couple of thousand brains donated so far?
Yes. So as I mentioned, we've sort of got two clinical cohorts donating to research. And so across those two, we've had now 2,900 donations, and 1,600 of those have been on the Parkinson's UK Brain Bank side. So a fantastic amount of people have sort of pledged to donate to research. But as research changes and the ways that we need to store tissue to make it useful for that type of research, there's always that need for people to donate.
And not just people with those conditions, but also family members as well, right?
Absolutely. And not just exclusive to family members. That is a very valuable donation. In often there'll be, say, if it's a daughter or a son, that will be controlling for some genetic background in a sense. If it's a spouse, that will be controlling for some sort of environmental impact there because you've shared a lot of the environment throughout your life. But so that's a helpful start. But anyone without a neurological condition of any sort, it's a very valuable donation to research.
Have you come across a donated brain from a Parkinson's family member that wasn't diagnosed in life, but subsequently you've discovered in your work that they had Parkinson's?
So Parkinson's disease is the most common manifestation of Parkinsonism. So in life, there may have been Parkinsonism. What we're really doing is finding out what the cause of it was. Was it Parkinson's disease actually, or was it one of those other conditions that may look like Parkinson's? Generally, if we get some brains from people who had no symptoms, we can still see the earliest signs of pathology. So those people are really useful to get research into the very start of the Parkinson's process. So although they had no symptoms in life, they had the early stages of pathology in their brain stems. So there's probably 5 to 10 years where you're developing pathology in the brain before you actually see symptoms.
What does that pathology in the brain look like with someone who's got Parkinson's?
So the most, I think everyone has heard maybe of the substantia nigra, the black substance, which is an area in the brain where there are cells which have this pigment in, which makes them look black. And they're really important for initiating movement, these cells. And these are the ones that seem to be hit really early on. And it's the loss of those cells and the loss of the substantia nigra cells that really underlies the movement problems, whether it be a tremor or whether it be rigidity. Any mix-up with the motor symptoms, it's probably, at least initially, down to those cells. But then, at the end of the last century, we actually found out what the protein is within those cells which has gone wrong. It's called alpha-synuclein. And we found that actually it spreads through the brain in a very stereotypical manner. And as it spreads, you start to see different symptoms.
And how do you see that? I understand there's some sort of discoloration that happens.
So when we do a brain dissection, which I do regularly every week to have a look and see what we're going to do in terms of the diagnosis, when I make a cut through the brain, the area called the midbrain, where the substantia nigra is, I can look at it straight away and see, is it still black? And if it's not, that's probably lining up pretty well with the clinical diagnosis of Parkinson's disease.
And does less black mean more advanced Parkinson's?
Yes. So, yes, there are degrees of loss. In fact, you need to lose about 60% of those cells before you get the symptoms. So again, it goes back to this, what I like to think of as a therapeutic window, where what we need to do is find out who's developing pathology and get in there with a drug that stops it before you get any symptoms. That's where we're all aiming.
It must be a real privilege looking at someone's brain. Do you look at it and think, just this piece of meat, or is it a human or what?
It's a massive privilege. People don't have to do this. And to hold effectively, I'm holding, 70 years of life experience in my hands.
Everything's in there, the loves, the losses, everything.
Absolutely everything. It's a record of their life. And, it sounds a little bit strange, but they are still a patient to me. They didn't have to do this. I treat the brain with the same respect I would someone in life.
I've never thought about it like that, actually, but you see a person, you see a life. You said you do this on average about once a week?
Yes, once or twice a week.
So
yes.
How many have you done, if I may ask?
A couple of thousand, probably.
Wow. I mentioned before we came on air, I'm due to have DBS shortly. And in fact, this episode might go out after my operation. I was asking you if you'd come across a brain that had the implants still in them, and you said you've had a few cases of that.
Yes, we have. So I think now I've seen 6 cases where when we're examining it, the electrodes are still in place. So in fact, that's really useful in terms of pathology as clinical audit. Where was it and did it work? So I can feed back to the neurosurgeons ultimately about exactly where it was.
Just explain that. How can you determine if it worked or not?
Well, this is all obviously recorded in life when you have the operation. Is it successful? Were there problems? Was it not quite right? There are a number of variables you can change in the surgery to make sure that it's treating the symptoms best it can. But ultimately, sometimes it doesn't work. And if we can look at post-mortem, we can see where it was and maybe work out why it didn't work.
Oh, so it's to do with the precise placement of the electrode.
Yes.
That might help answer questions as to if it was effective or not.
Yes, absolutely.
When you've made breakthroughs here, what are the major things that you've discovered about Parkinson's brains in this place?
Well, obviously, we are a service. We're finding the brains. We're diagnosing the brains. We're characterizing them, everything that's gone on in these brains. But it's our role to get them out there, the tissue out there, to as many labs worldwide to maximize the chances of progressing and working out how we're going to stop this. We do our own little bit of work. We obviously, I'm still very interested. I see so many brains. My particular interest is what's the relationship between the pathology I see down the microscope and what happened in life. And can we dissect the processes a little bit? But all sorts of things are going on. We've found out by looking at human tissue that there's maybe more than one type of the alpha-synuclein protein. So, for example, I'm sure you're aware of the condition multiple system atrophy, which may look like Parkinson's disease. It's the same protein, but it's targeting different parts of the brain. And in a dish, it's acting in a different way. So almost we're getting strains of pathology. Not everyone's the same. And again, you can recreate this in a dish in a lab with recombinant or synthetic proteins. But ultimately, it's what happened up here in the individual. So again, it's vital to have that human tissue to actually work out what's going on.
So you can see different types of Parkinson's, MSA, PSP,
yes,
and Lewy body dementia?
Yes, so Lewy body dementia is, again, alpha-synuclein is the problem. That's the protein that's gone wrong. PSP is slightly different, and it's the tau protein. It's a different protein that's causing the problems.
MS, does that look totally different to Parkinson's?
MS, yes. MS is a demyelinating disease. So essentially, you've got in your brain, you've got the nerve cells and you've got all the wiring. And in MS, the insulation on the wiring is lost, which makes it less effective. So people will either lose function or perhaps reduce function, depending on where the pathology is.
What could you do with more brains if you had more donations coming in? Or let me put it another way, is the lack of brains being donated a limitation to you in any way?
With research like this, especially when we're looking for minor differences between the same disease type and things like that, more is better. It's that sort of big data approach, plain and simple, in a sense. But what we're particularly interested in as well, as Steve was mentioning, is having individuals who may not have Parkinson's, having them donate as well, because what we're looking for there is that early stage, and that's what we're trying to understand, that early part. But more than that, when researching Parkinson's, there's a lot of interest in this substantia nigra area, and people may look at other areas of the brain, but they'll always want to coordinate that with what they're seeing in the substantia nigra. It's a very small area, and everyone always wants to reference it. And so what you get there is even individuals who may say, not really bothered about that, I want to look elsewhere in the brain and try and understand what's happening there, they're only going to be able to do that if there's a donation or a brain where we still have that substantia nigra.
And do you find there, in certain groups of people, there is reluctance to donate their brains, either for ethical, religious, or other reasons?
Absolutely. There's a wider issue, I think, with different groups wanting to access the medical services. So that's sort of what we are working against as well. When sort of approaching different communities that have, say, historically been against body donation, that sort of thing, there's been a lot of work by the NHS organ donation scheme to sort of help break down those barriers. And so what you tend to find is minority groups aren't so much against the idea of brain donation explicitly, but they're just not engaging with the medical service in the first place. So we're sort of coordinating with other groups to make sure that first barrier is broken down, and then subsequently that sort of engagement with things like brain donation will come further.
What's the process of donating your brain then? You say you fill in a form, but what actually happens when you get it taken out and stuff? Is it
Of course. No. It's done by trained professionals who absolutely know what they're doing in that sense, so don't worry. But everything happens very quickly, in a sense, and everything will occur locally, which is sometimes what people get a little bit confused about, as we're based in London, but we're able to facilitate donations across the UK. That'd be Northern Ireland, the tippy top of Scotland, down the bottom of Cornwall.
Very good. And you say it happens quickly because it has to happen quickly, right?
Absolutely. And so what we're looking at is things in the brain that over time degrade. And so we have to make sure that the tissue is as viable as possible when it gets here, so that when we preserve it for time onwards, it's in a good condition to do that. What we find is one of the major factors to this is time from when the person passes away to when we've been able to preserve those tissues, whether that be in something called formalin or whether that be freezing the tissue. The sooner we do it, the better and the more useful for research.
And what is that window? How long is that?
So that's 48 hours.
48 hours.
And unfortunately, the reason we can't accept donations beyond that, as I say, is because the tissue begins to degrade after that and isn't viable for research anymore. And we basically have ethical permission to collect donations for research.
And going back to Dave's question, without getting too gruesome about this, how do you physically get a brain out of the head?
It's pretty much as you might think. The top of your head is like the top of a boiled egg. You take off the top of the boiled egg, much as you would when you're eating a boiled egg. But I should reassure people that actually by making incisions in the scalp, reflecting the skin, removing the top of the skull, and rebuilding afterwards, you wouldn't know we'd been there.
That's it. And that's a main concern for a lot of families is, you know, my family are going to want to see me and that sort of thing. And I'm worried that I'm not going to look how I should. And just to sort of put that to bed in the sense that that's absolutely not the case. These are trained professionals doing this procedure. They know what they're doing. Similarly, funeral directors who will coordinate with those onward arrangements know what they're doing as well.
So your eyes and ears and everything will be in the same place?
Absolutely.
Yes. Absolutely, yes.
But these are considerations that family members have, right?
Yes.
Presumably. And you say you work a lot with funeral directors. I was talking to you the other day, Rory, and you said you were taking a delivery at the weekend. And by delivery, you don't just mean a DPD delivery, right? How does it actually come if I can...
No, absolutely. And that's it. So what we try and do, because obviously at this time, someone has made that very selfless pledge to donate their tissues to research. And that's a big deal. And obviously at the time that they do that, they've passed away. And for families, that's a very stressful time. And so what we try and offer as a research service is we look after that process as much as possible. So we have an emergency phone number that can be called any day of the week. And they call us. They let us know, we take some details at that point, and then we look after the rest of the process. And so when someone may pass away in the community, the first step is that they go into the care of a funeral director, and they'll rest there basically until certification of the death is complete. And that's done between the GP surgery and an independent doctor who will basically make sure that they agree with what they're proposing as a cause of death. So that all happens. I act as a professional pesterer, essentially, in that whole process, making sure people do things as quickly as possible. Once we have that certificate, that's when we are allowed to make that transfer from the funeral director's care to a local hospital. That'll occur wherever the individual's passed away, then the next major trust that will basically be able to support these donations. And from that point, we'll do that all within 48 hours. And once the donation is done, we'll enable a courier to take those tissues from there to our lab in London.
And are there equivalent brain banks outside the UK? Are there international ones in other countries?
Absolutely. There's equivalent brain banks outside the UK. There's equivalent brain banks inside the UK as well. And so we work collaboratively with a lot of other brain banks, but we are the specific brain bank for Parkinson's and for MS.
And you mentioned, Steve, earlier that some of the samples go to researchers outside the UK. Is that right?
Yes, we have a review committee. So researchers anywhere in the world can say, can we have some tissue, please, to do this, this, and this? It's looked at by the committee. If it's deemed to be a useful use of the tissue, we can dispatch tissue all around the world, yes.
And why do you need to go through that qualification process? Is that because you've got a limited number of tissues?
Obviously, yeah, supply and demand is part of it, but actually we need, because we give the ethical permission for them to use the tissue, we need to make sure it's a good use of that tissue, and that's why we have the review committee. And
that's the real trick where, as I say, when people visit the brain bank, is we're not a collection. We don't collect these donations to put them on a wall forevermore. We're thoroughly involved in making sure we get these donations to use in research and make sure the researchers who are doing that work actually understand how to use the tissue and be able to do it. So we act as an element of expertise there, but through being a research tissue bank in that sense, we can act as a mini ethics board. And so we have that privilege, but obviously we also have to make sure that we are reviewing each request as it comes in. And that's with a group of scientific experts and also lay members, both from both charity sides.
People really wonder how you treat the brains, and it seems treated very well. You know, you're saying what a privilege it is to be inside someone's head and see a brain. You can guarantee they're going to be looked after, these brains.
Yes, as I say, this is an individual who's made this bequest, and they're treated here as an individual. So some of the tissue won't be used because it can't be used, but it's all kept isolated, that individual. It's not just, oh yes, that's not used, let's just throw that away. Everything is respectfully disposed of by incineration, and it's for the individual.
And just going back to the demographics of the donations that you've received so far, in the case of Parkinson's, I presume it's mainly older people that you've taken receipt of older brains, as it were. Are you missing a piece of the puzzle in terms of people like me and Dave that are young onset? Is there value in us encouraging our fellow members of the young onset community to donate their brain? Absolutely.
And that sort of comes back to that sense of we need to understand every type of Parkinson's there is. And so we strongly encourage everyone who feels that they're able to donate in that sense. We struggle a lot, especially with sort of representation from minority groups, as we touched on earlier, where you're spot on in the sense that we have a large amount of elderly donors. We also have a large amount of white British donors, and that's, you know, a strong majority in that sense. We've touched on the fact that there is sort of cultural barriers there, but for a lot of people, they're just not given the information in the first place. And so we always strongly encourage anyone who feels that they can donate to sign up to donate.
What amazing things have you found in people's brains?
Well, obviously, we find the pathology that we expect in terms of the Parkinson's disease, but what we also do, we're part of an audit because we need to know everything that went on in that individual. So sometimes we find things that weren't picked up in life. So, for example, sometimes I find tumours, which may or may not have had any symptoms in life. But obviously we have to make sure we've documented that. Sometimes people have strokes that weren't picked up. And I can pick up where there's an area of the tissue where there's been a stroke because the tissue's died. And again, that may have contributed to the symptoms in life. So this is why we have to do a full audit of the whole brain. I have to look at it in detail all the way through and find out everything that happened.
And this is it. This is what I always like to say, and pathologists like to mention it, is I think, the buck stops with the pathologist. That's where that definitive diagnosis is. Neurologists are fantastic. I'm not looking to cause any, stir any trouble here. And they can diagnose these things in life wonderfully. But that diagnosis isn't actually complete until we've looked at that tissue. And that's sort of what we're able to provide.
So the only surefire way of diagnosing Parkinson's 100% is, as a result of what you guys do, seeing it after the fact, after death, by looking inside the brain. Is that right?
Absolutely. At the moment, autopsies are not generally needed for the rest of the body because imaging is so good that you can be fairly confident with the diagnosis in life, particularly if there's been interventions. But with the brain at the moment, we don't have imaging of the resolution required to be able to tell you what the actual final diagnosis is. So at the moment, Parkinson's disease has to be confirmed at post-mortem.
I was going to say, scanning technology is getting better all the time. Will there come a time when you don't need brain donations, when you can just do it all electronically?
Oh, AI, here it comes. We're a long way off that at the moment, because all the changes that we're looking at and defining the diagnoses are at the cellular levels. Small amounts of protein. This is very, very small. This is down the microscope, literally. And at the moment, no. In life, we just don't have the imaging that can discern that type of change.
That's it. And I think, not to speak out of my place, I know I'm not the research one, but what is happening with the tissue that is donated is it's that really, I hate the phrase, but it's that basic research. There's nothing basic about it in the sense that it's that true understanding of what's happening at the cellular level. And it's from that we can build on top of that to say, then we say, we know that this is what's happening at the cellular level. We know how that matches with scanning and that sort of thing. And that's how we can bring those together. But sort of talking about earlier with the DBS probes, that's sort of what we do here is we know DBS probes work. Why are they working? And then there's been some work into that to say the mitochondrial activity is going up around the DBS probe. Why is that happening? And it's those sort of stepwise questions that we can only do by looking at the tissue. We're not going to understand that from the imaging.
Right, so we're going on a little walk, aren't we?
We are indeed.
Excellent.
So we're going to be walking down from the fifth floor down to the fourth floor, which is where the brainbank is based. And so, hopefully what we want to show you is just a bit of that journey of the brain donation. So we touched a little bit on about how we work beforehand to have that donation done. But then obviously once it's received here at Imperial College, that's when the work begins to do the diagnosis, the reporting on that case, and then the subsequent passing on to research.
Right.
So it's walking in here, and what we'll be doing is we'll head into, we'll normally receive that brain downstairs, as you mentioned, working on the weekend.
So it literally comes into reception, does it?
It does, yes. Normally we'll meet them if we'll be kind enough. And then...
I presume it's not signing in.
Correct. And then we receive that, and then the work begins here.
Right.
So I obviously receive the calls at any time, but the technical team have to be here at any time to help do that donation.
Right.
So we're in the dissection room now.
And it's noticeably cooler in here, right?
It is. And basically what Steve's going to do is walk us through that first step. I believe this case is a whole fixed case. And what that means is with that 48-hour time limit, we weren't able to get it to London within that 48-hour time limit. So we had to ask the hospital to fix it in formalin on site. And so the whole brain was fixed in something called formalin so that the tissues wouldn't stop being viable.
Is that like freezing it? Or?
No, so it's in a liquid. So it's in room temperature. It's what you'll find in any pathology lab across the country in a sense. What we ideally do is receive a brain here at the lab, and that way we'll take half to be frozen and half to go into formalin. But we'll always have to do this diagnostic step, which is what Steve is doing.
Right.
You're more than welcome to participate in the dissection as much as you like.
Wow. Did you hear that, Dave?
And so we have these gowns if you do want to sort of hold the brain or investigate any pathology and that sort of thing. And we've got some gloves over there.
Send me a bucket with a brain in it.
Yes.
There we go.
Wow.
Incredible.
Amazing.
Thing of beauty, isn't it?
Saying earlier,
Wow.
every brain is different.
What does that weigh? Like 2-3 kilograms? Or?
No, it's average adult male, female, about 1,400 grams. 1.4 kilos.
Okay.
Yeah, what's that in old money? About £3?
About £3, yeah.
We
Yeah.
don't talk in old money. It's scientists, Steve.
So what I'm doing here is I'm going to have a look at the whole brain, see if I can see any signs of changes
On the exterior.
on the exterior.
What is it you're looking for?
So here I'm looking for any signs of infection.
Mm-hmm.
Was there an infection near end of life? Could it have contributed?
Mm-hmm.
There's no indication of any infection here at the moment.
Mm-hmm.
I'm looking at any parts of the brain slightly smaller than they should be.
Mm-hmm.
And I have to say, in this case, the front of the brain
Mm.
is a little bit small.
Yes.
Right.
And that is not unusual for someone that's had Parkinson's for a long time.
I see.
Maybe later they might start having a little bit of problem with decision making,
Right.
things like that.
Cognition.
And really it's your frontal lobes that are the
Right.
seat of your personality. So when there's a little bit of change there, that's what you might see in life. I'm feeling the brain. Are there any softenings? Because that might indicate there might have been a stroke. And again, in some people, that may be what actually is end of life, might have caused death. So we need to have a look there. You can see here, we have the blood vessels that supply the brain.
Oh.
And by looking at those blood vessels, I can see if there was any vascular disease. Because, as we just said, it's about 1.4 kilos, the brain. So it's quite a small proportion of your body weight, but it takes 20%, a fifth of all of the blood from the heart goes to the brain. It's very demanding. We just remove what's in the way here. I'm just going to remove the blood vessels. And what we're actually looking at is the brainstem under these vessels, just here.
So that's what connects to the spinal cord?
So there,
Yeah.
that was the spinal cord.
I see.
So this is at the
Back.
I see.
bottom of the back of your skull. Here.
Right.
What you can't feel here is I'm cutting through these blood vessels, and they're quite tough. So the old hardening of the arteries.
Right.
This is because this gentleman had some atherosclerosis, which is a build-up of fat in the blood vessel walls, and it hardens and it makes the vessels less compliant. So in normal function, your blood vessels are always opening and closing, dilating, constricting, depending on what blood flow is needed. When you get a build-up of the fat inside the vessel walls, it makes that less effective. So,
Is that diet that would cause that?
yes, diet, in terms of vascular health, there are three things. It's all very boring, I'm afraid, in lifestyle. It's don't smoke, balanced diet, and plenty of exercise.
Right.
They're all helping ultimately to reduce the risk of degenerative disease in old age, but really what they're doing is just keeping the heart and the blood vessels healthy.
Right.
I like to think balanced diet is open to interpretation. The
Yeah.
other two, not so much. Don't smoke. But the other one,
Yeah.
So here you can see, if you look at that vessel, that's one of the big vessels that goes up the side of your neck, the carotid artery.
Right.
The internal carotid goes on and supplies the front part of the brain. This is really quite rigid,
Right.
but it's not really blocking the vessel. So that's the worry, is when you get a build-up of that fat
Yeah.
too much, it stops blood flow.
Right.
Not evident here. So now what I'm going to do is I'm going to cut through the midbrain, and this will be our first indication as to whether or not this was Parkinson's disease.
But as it's come to you, it's a Parkinson's patient.
I get minimal actual clinical data here, pretty much what was passed on at sign-up usually, which is probably good because I then don't end up looking for what I think I should find.
Oh, right. So you're not biasing
Yes,
yourself.
yes,
Right.
It's better to just look and find what you find.
Right.
So I'm going to make this cut, and
Okay, so this is the midbrain. So we had the spinal cord was down here.
Yeah.
We have the medulla, the pons, and the midbrain, which is the top of the brainstem. And what we're looking at here is what, in this case, remains of the substantia nigra.
Oh, right.
Because there should be a jet black line across there and across there. But in fact, all you've got is a little bit of the pigment left towards the midline here on either side. So this is completely consistent.
So this should be black.
It should all be dark. Yes, black.
So can you tell the stage of progression from the...
It's not an exact science, but
Right.
with that degree of pigmentation, loss of the black substance,
Yeah.
I think at least 5 to 10 years.
Wow.
Yeah.
And just to help listeners visualise, we're looking at something that's about size of a...
Don't have coins anymore,
No,
do
we don't, but
It's a 50p piece.
50p piece. Yes, that's a good analogy.
Yeah. So,
Yeah.
and
There's the two sort of V black lines that should be jet black, but they're looking more grey here.
And just a dark around the edges,
Yeah.
aren't they?
yes, that's it. Yes.
But they're more grey, right?
Yes.
Okay.
So, first pass, this is consistent with what we might see with someone that had Parkinson's.
Right.
What I'm going to end up doing here is cutting the brain up into a series of slices such that I can look at all the key areas and see if there's anything I can see, and to sample those areas so that we can look at them later down the microscope.
I see.
So that's what we do here. And to do that, I'm going to have to make a cut right the way through the middle there.
Wow.
And then we'll see what you may have seen on imaging. I'm sure you have if
Yes,
you're going to have the DBS.
Yes.
So I shall make this cut.
That looks a very sharp knife.
Yes, it's not a bread knife.
Wow.
And here, this is perhaps what you're more used to seeing
Yeah.
on the
Yeah.
image.
You scan, it looks like that, doesn't
It
it?
Yeah.
looks like that, yes. So what I can look at here, first of all, as with all things, you're looking for asymmetries, because the left and right side of the brain are pretty much exactly the same structurally. So the first thing, does it look symmetrical? Is there any movement of the midline? Could there be something over here that's pushed the brain across the midline? So you're always looking for asymmetries.
Can I say, from my view, it looks pretty symmetrical.
And you'll be glad to hear I agree.
Good.
Yes, pretty much exactly what we'd expect.
Right.
What I would suggest is that the ventricles, which are the holes in the core of the brain there, are slightly bigger than normal. Okay, but this gentleman was 86 years old, so I'm not surprised.
Right.
So now I'll show you the slicing technique so that we get a ½ centimetre slice. And we need them to be that thick because some of the areas we need to look at are really quite small. And if we had thicker slices, we might miss them. So that's why we do these slices. So,
And those ventricles that Steve was talking about there, was talking about how we're not just a brain bank, we're a tissue bank. So that what is flowing through those ventricles is CSF, cerebrospinal fluid.
Okay.
And that is what we will also collect to sort of do some analysis of
Fluid.
that fluid around
Right.
the brain. Yeah.
I mean, the old cliche is like slicing a piece of ham, right? I mean, it is that.
yep. So
Beautiful slither of brain there.
It is actually quite pretty,
It's
isn't it?
gorgeous, isn't it? Yeah.
it is.
Amazing.
I mean, look at the symmetry on that. That's wow.
It is. It's an amazing piece of machinery.
Right.
So you're going to have a procedure.
I am. My DBS is coming up shortly. Are you going to?
I can maybe point out to
Please
you
do.
where we're thinking
Please
about.
do.
So remember we cut the brainstem
Yes.
here. That's where the substantia nigra is.
Yes.
They're not going to stimulate the substantia nigra. They're going to stimulate areas quite close.
So it's
Red one.
below it or above it?
Above it.
Above it.
Above it. So this structure here in the midline is the thalamus. And think of the thalamus as a telephone exchange. All the information coming up from the body and the instructions going down go through the thalamus. And below the thalamus is the subthalamic nucleus.
Right. Otherwise referred to as SDN,
STN.
which is the most common target area for DBS.
Correct, yes. So that's where they're aiming. And there it is. It's a small area.
That's tiny.
It's tiny.
Oh my word, that is tiny.
It's maybe 4 or 5 millimetres.
Wow, I mean, that's not even a half penny, is it? That's
No.
That is tiny.
Yes. So they're going to get the electrode. It'll be going in about here, slightly far forward. It's going to go down through the brain, causing minimal damage. It's really amazing how they can get it there without causing any other problems. And that will sit in there on both sides for you. And one assumes it's for your tremulous.
Tremor, stiffness, slowness.
Yes. So it's all part of the circuitry that initiates movement. And what you're doing almost is resetting the circuitry. Because it's out of sync, because you've lost those nigral cells, stimulating in this area seems to readdress the balance. So that's how it works.
Wow, fascinating. And you pointed out the one target area for my DBS SDN.
Mm-hmm.
Could you point out the other target area in my case, which is GPI?
GPI, yes.
So it's even smaller than the SDN.
Yes, it's not far from it. So here, these triangles are part of the basal ganglia. You may hear this term. This is where the substantia nigra neurons were projecting to. They were connected to this area, and this is what was controlling, getting up out of a chair, whatever, controlling that movement. And these are vital in that. And these 3 different structures in those triangles, they see they're interlocking triangles. The big one on the outside is called the putamen. And then you've got the globus pallidus external part and the globus pallidus internal part. That's the smallest triangle right there.
Is the eye in GPI internal?
Internal, yeah.
Right.
Globus pallidus.
That is tiny.
It's that bit there. So not that much difference from the STN.
Yeah.
And very close, obviously.
And just, if you can, Steve, just explain why, in my case, they're going to be targeting the GPI, if you can. I'll give you a clue. You could probably see it in me right now. Yes.
Disco.
My disco, as we call it.
This is to help control unwanted movements.
Yes.
Dyskinesia.
Dyskinesia.
Yes. So these are movements that you have no control over.
Yes.
So this is, we hope, what's going to give you back control.
Hopefully. Wonderful. I should have asked you this at the beginning. How many of you are there at the Brain Bank?
That's a fantastic question. There is sort of 6, sort of 8, sort of a lot more than that. So the core team, the core technical team is myself, the tissue bank engagement manager, the tissue bank manager, and then 4 technicians who look after 1, this sort of process, 2, that tissue culturing process, those sort of cell lines, and then we have 2 technicians who are looking after liaising with researchers both on the Parkinson's and the MS side.
I see.
You could then open that up to the man who's been talking all the sciences now, the scientific director, and we have a scientific director on both sides.
Yeah.
And then beyond that, we also work with neuropathologists across the country who assist us in reporting on these cases.
And you're funded by both Parkinson's UK and the MS Society, is that right?
Yeah, absolutely.
But you're both working, your employer is Imperial, is it?
Yes.
Right.
Yeah, and that's it. And so it's really quite unique at the moment, especially in a time when things are not being funded as much as possible and that sort of stuff. But to have this 3-way partnership where the infrastructure is provided by Imperial, and you've got the processes being overseen by the charities and funding that process, we're a lot better placed to accept a lot more donations and to continue accepting a lot more
Could you exist without those charities?
No.
As simple as frankly. Yeah. Without the donations, 1, of people donating their brains to us, without the donations, 2, of people donating to the charities, this work wouldn't happen at all.
Well, you heard it there.
Yeah.
I must say, listening to both of you seem very passionate about this. It must be very rewarding to do, right?
Absolutely. I mean, the days can be long and all that sort of stuff, but I never go home not feeling like there's a purpose and a reward from this work. So it's very good.
It's the other way around, yes. I actually want to come to work in the morning.
Oh,
yes.
Wow.
Yes.
I just find it fascinating.
It is.
And if we can learn something from this, that's why we're all here, isn't it?
Absolutely.
Amazing, thank you.
Nothing to see here. Nothing to see here. Nothing to report. Nothing. Just ignore it.
That was very strange, Kuhan. I've just beaten you 6-0.
Yeah.
But I feel it feels a bit of a hollow victory because it was like you were relearning the game.
Well, to be honest, that's what life post DBS has been like. I have to relearn things, and it's really odd. Couldn't hold the racket properly for some reason. Can't figure out why, but I'm trying to be patient and yeah.
I think you might have to get a lesson just to relearn the skills that you had before because
just a grip specifically.
Yeah.
I think I was slicing it a lot. I was hitting down onto the ball, whereas other than that, I think I can feel my movement is okay, but just something about the grip. And I found other things in life, using a knife and fork, putting the seat belt on, things that I normally use the other hand for. Now I'm having to go back and use my correct hand. I'm having to sort of do it in the normal way. Whereas I think after years of various different symptoms, some of which still exist, your mind and body adapts, doesn't it? And you have to kind of relearn now.
Exactly. You have to relearn tennis. It's going to be quite strange, that, because I really thought you'd stop back into it and your movement looked good, but you weren't throwing the ball high enough. It was as if you were still restricted by the Parkinson's, and you're not. You've got more movement now.
I mean, look, I put my hands above my head quite freely now.
It's quite weird. You're doing your old movement without using new movement.
Yeah.
Very strange.
You asked, when we had Laura, the physio,
you
might come and watch us play tennis one day, actually. You asked it, is there a concept of sort of rehabilitation post-DBS? Not sure anybody's sort of offering that. In the same way that rehabilitation post-stroke or something like that, where you're relearning to do stuff the correct way.
Normally after a big operation, you have some physio or something, don't you?
Yeah, exactly.
You have a course of physio. When I had my knee done, I had a new half knee. I had a course of 10 weeks of physio afterwards. And the physio taught me to move again and get the movement back. Now you've got more movement, you maybe need to harness that.
Exactly.
Maybe need someone to show you what you can do now. The motor skills, you've got to get them back again.
Yeah, exactly. It's strange. I need to be patient, don't I, Dave?
You get very frustrated during the game.
Yeah. But that's just me, and also it's also a reminder that actually, I don't think this is how it's going to be all the time, but it is a reminder that DBS is not a cure.
No.
And it is not perfect. And there might be types of movements that I'm more restricted in than previously. Another step on the Parkinson's journey and another reminder that nothing is perfect. Disease still progresses and, got to continue to remind myself. And maybe this is a healthy reminder. It's also very early days for me, right?
Yeah.
Still haven't had all my settings tweaked.
5 weeks after your operation, we've just had a game of tennis. It's quite incredible, that.
Don't say 5 weeks, Dave. I'm not meant to play. I'm not really meant to be playing until next week.
Oh dear, sorry. I made sure I didn't hit your head though.
Yeah, it was quite a leisurely game. You meant to give it 6 weeks, but I don't think that was a factor in my loss today. But
you don't be headering it, do you? Really?
No. Quite an interesting experiment. A clinician sees me playing tennis. I think they could decipher a lot more from both of us, actually, from any two Parkies playing tennis or everyday activities like that compared to some of the standard neurology tests. But anyway, that was a nice visit to the brainbank, wasn't it? If you hadn't worked it out, that was before my operation. But got to see the target areas in the brain where, well, put it this way, I understand now why they call it deep brain stimulation, because it really is the most remote part of the brain, right?
It was incredible seeing him dissect a brain. It's quite moving, actually. Looking at something and thinking all the emotions, all the loves, the losses were in there,
And the way that they treated the brain like a patient.
So gently,
So gently.
Yeah.
The way that Steve was describing how he treats a patient with respect. Yeah. Still thinks of him or her as a patient. It's really nice.
Incredible. The gift of a brain could eventually find a cure for us. So, if you want to do it, in the show notes, it shows you how to do it. And they said as well. So.
Yeah, fill out a form. There is a form to fill in. The link is in the show notes. And well worth it. You never know what can happen. And they were saying how people, when they send the form or when they end up donating the brain, they maybe send a poem or something, as a, as a marking the event, as it were. Point of acceptance, I think, almost, for a lot of people that, because it's a big step, right? You don't want to take it lightly, donating your brain, but...
No, it's a big thing, isn't it? Because you feel like, your body being separated from your mind is going to be quite a weird thing, but trust me, they're gentle with it, they handle it with care. It's beautiful the way he treats that brain.
Yeah, it was...
Really
I was really quite surprised. I thought it'd be a bit more like being at the butcher's, but nothing like that. Nothing like that at all.
And the slice he took of it, so thin,
I know.
Such an art.
Yeah. Sorry, it sounds strange, but when you see it, it's so pretty. The symmetry, the kind of like the shapes, the patterns.
It's a beautiful thing, isn't it? A
Really-
beautiful thing, the human
Really
brain.
is, really is. I thought it was really sweet also when Rory kind of had this fanboy moment with you. Do you remember when with the darts? He recognised you from the darts, and I think you told me this before. In Australia, darts is huge, right?
Yeah.
And didn't he show you his own?
Brought his darts in to show me,
Yeah.
He plays darts and he had his own set of darts. He
Yeah.
showed me them,
Well, as in his partner had sort of done some art on the.
He's got his own flights, yeah.
Just explain what flights are, Dave.
Flights are the bit that you stick in the top of the dart, you know.
Right. Not the pointy
coloured bit, not the pointy, on the other end.
And people kind of personalise their.
Yeah.
Right.
Phil the Power Tailor has power on it, you know.
Okay.
All that sort of stuff.
Right. So yeah, it was really sweet actually, and obviously recognised you from way over in Australia and way back in the day, I guess.
Speaking of which, you've been recognised a few times, but someone else.
I have actually.
You've got a doppelganger somewhere
Yeah.
that keeps getting people walking up to him saying, "Are you something to do with Parkinson's?"
This is quite unbelievable.
It's happened twice, hasn't it?
Well, it was the same occurrence actually. It was the other person in the story. Let me just read this story to you. This guy sent me a message on YouTube the other day. Hello, Kuhan. Today I was approached by an unknown lady in central London. Okay, cool. Today I was approached by an unknown lady in central London. She asked if I was involved in Parkinson's. I said I wasn't, and then showed me a Google image and commented on how similar I looked to you. And hence to her query, I have to say there is a resemblance. So this means you are extremely handsome. Ha ha ha. On a related note, I knew Tom Isaacs, so...
Guy who found a cure Parkinson's, yeah.
Exactly. No longer with us, unfortunately. So I do have a connection with your noteworthy cause. Tom was from my school, but we were in different eras, and I supported a few of his charity events, which involved professional cricketers. Wonderful. I think that's from Rajiv, who's huge.
Kuhan too.
Well, apparently, I did. Did I show you a photo of him? He does.
No.
He does look very identical to me, I've got to say. So I didn't think much of it, because I don't know. People reach out when it comes to that sort of thing. I'm always kind of wondering where they're coming from. So I didn't actually reply for my sins, but I will today because I got this message the following day. Hi Kuhan, my name is Anna. Having supported my mother as she lived with Parkinson's, I am now an active volunteer with Parkinson's UK, involved in many different roles. Good for you, Anna.
Well done.
Yeah, lay grant reviewer, involved in the steering committee, and a Parkinson's cafe host. Been inspired by your story, listened to your podcast and read about the app you have developed. I wanted to share an experience I had yesterday. I was at the Welcome Collection on Euston Road. As I came out of one of the exhibitions, there was a man sitting on a sofa. Thinking it was you, I thought I would introduce myself and asked if he was Kuhan. He laughed and said no. A few awkward moments passed. And I explained that he looked like someone I knew of and had not wanted to miss the chance to say hello.
Oh.
Very nice, right? He then laughed and said it was obviously his good looks that had caught my attention. More awkward moments. I then explained that Kuhan and I were involved with Parkinson's UK. When I mentioned Parkinson's UK, he asked me if I knew Tom Isaacs, as he'd been at school with him and played in a charity cricket match that Tom had organised years ago. I said I'd met Tom Isaacs, but I was involved with Parkinson's UK, not Cure Parkinson's. Both great charities, by the way. Again, the man laughed that I'd mistaken him for you. I said, look at the two Parkies in a Pod webpage and you'll see why. He asked me if you were Sri Lankan as he is, and then Googled it on his phone. His jaw dropped and he looked at me. He said he knew exactly why I'd been mistaken. The man is your doppelganger. I felt relieved that he had understood why I'd mistaken him for you.
Very great story.
Yeah, very strange meeting and shows the world of Parkinson's is very connected. Very true, very true. I left the building thinking that the thing about 6 degrees of separation was proved at that moment. I had to share this with you.
Oh, it's brilliant.
I hope to actually meet you one day, perhaps at Parkon this year. From Anne. Thanks, Anne. And what a sweet story. By the way, we are going to Parkon, aren't we, Dave? Me and you.
We think we're going to do another live podcast from now.
We are. Parkon is happening at the end of October, 24th and 25th of October in Birmingham this time. And I think there's slightly more capacity than last year in person. And if you can't make it in person, you can join online.
Yeah, if you want to come along as meet as well, tickets are available now. So
Yeah. Tickets meaning it's free, right? You just need to
register.
register. We'll leave a link for that. What an amazing story, right, Dave?
That's ridiculous.
It's ridiculous. Yeah. Do we have any people doing amazing things today, Dave?
We've got a few, yeah.
Okay, come on.
Got a few things.
Let's hear it.
People with Parkinson's do amazing things.
Okay, Jimmy Choi, alias Fox Ninja, returned to compete in American Ninja Warrior TV show. Jimmy has had Parkinson's for more than 20 years and uses his fitness to fight the condition. This is how he got on. He did brilliantly.
My father has Parkinson's, and Jimmy Choi has really not only inspired me, my family, but really just shown the entire community that with Parkinson's, there's still a lot of hope and a lot of life.
There's just no quit in this man. He raises money and awareness for the Michael J. Fox Foundation. You can see the Fox Ninja on his shirt.
I tell you right now, he gets through log runner, Matt. That's the win there. For so many reasons, and people who suffer with Parkinson's, they know you start to do the shuffle. It can be hard to pick up your feet.
Jimmy Choi used to defying the odds.
Come on, just do it. I think he's thinking about it. He's just got to approach it.
Here we go.
For one moment in time, just go for it.
Sweet moves. Jimmy Choi.
People with Parkinson's do amazing things.
Food blogger Adam Roberts, the author of Amateur Gourmet, has gone public about his Parkinson's. He says he'll do everything he can to keep writing, cooking and playing the piano. You can read his Parkinson's blog in our show notes.
Wonderful. Who's that, Adam?
Adam Roberts is the Amateur Gourmet, it's called. He's big on social media.
People with Parkinson's do amazing things.
Finally, we've got to say farewell to the great Ozzy Osbourne. What a way to go out. 40,000 fans turned up to say farewell to Ozzy at Villa Park. Black Sabbath played the final gig. The Prince of Darkness sang his heart out from a special throne. He absolutely nailed it and then passed away a couple of weeks later. What a way to go. We'll leave you with a bit of Ozzy.
Oh, okay, cool.
Words of wisdom from Ozzy.
Yeah, are you going to sing us? Is this going to be a live rendition from you, Dave? Or?
No. See you soon.
See you next
Leave you with Ozzy. Bye.
Cheers. Bye.
Your support over the years has made it all possible for us to live a lifestyle we live. Thank you from the bottom of our heart. I love you. We love you. And it's a song called Paranoid.
You've been listening to Two Parkies and a Pod. If you've enjoyed listening, please do rate, review and subscribe to be alerted to new episodes. And it helps others discover the podcast. If you'd like to get in touch, you can send a message to hello@twoparkiesandapod.com. That's hello@twoparkiesandapod.com, or reach out to us on social media.